All right, next up we want to talk through the actual process of meiosis itself. So we're going to start off by talking about the two phases of meiosis, Meiosis I and Meiosis II. As I've pointed out before and will point out again, scientists are not terribly clever when it comes to naming things. Almost everything is named for either the order in which it was discovered, the order in which it exists, or it is given a descriptive name that just happens to be Latin or Greek for what the thing actually is. Like a hydrophobic molecule, meaning that it is afraid of water. It's nonpolar, meaning it has no poles. It does not have a positive charge or a negative charge. Scientists are very, very descriptive with their naming and not terribly creative, which I think is fantastic. I think it really, helps us understand, helps us learn, helps us memorize. So, phase meiosis occurs in two distinct phases, Meiosis I and Meiosis II. DNA replication occurs before Meiosis I, so in the S stage of interphase, just like we have in mitosis. The very first thing we have to do is replicate our DNA. DNA replication is going to occur before Meiosis I in the S stage of interphase. Both Meiosis I and Meiosis II contain the stages of prophase, metaphase, anaphase, and telophase. And they are going to be very creatively called in Meiosis I, prophase I, metaphase I, anaphase I, and telophase I. And in Meiosis II, they're going to be called prophase II, metaphase II, anaphase II, and telophase II. So we're going to start off with prophase I of Meiosis I. In this very first stage, we're going to see the spindle form. The nuclear envelope is going to fragment, and the nucleolus is going to disappear. All of this is exactly what we saw in prophase of mitosis. We're going to see the spindles form, we're going to see the nuclear envelope dissolve, the chromatin is going to condense down. However, in prophase I of Meiosis I, we are going to see something a little bit different happen. We're going to see the homologous chromosomes start to pair up. This is going to be the synapse, and like we said earlier, this is where we're going to get a little bit of genetic variation during our crossing over event. Synapsed homologs are referred to as a bivalent, just means that they have two homologs, or they'll also see them referred to as a tetrad with 4 chromatids. This final bullet point you don't need to memorize. I'm not going to quiz you on that or ask you about bivalents or tetrads. I'm just giving you some more information for when you're reading this on your own or if you're looking up your own tutorial videos. Next, we move into metaphase I. In metaphase I, the homologous pairs are going to be arranged at the metaphase plate. Now, their arrangement is completely random. We have no idea how they're going to line up. They could line up maternal-paternal, maternal-paternal all the way down. They could line up maternal-paternal, paternal-maternal all the way down, every other sort of thing. The way they're going to line up down this metaphyseal plate is called that independent assortment. With that independent assortment, I'm not sure I said this last time, but all that really means is just because you got your mom's chromosome for finger length doesn't mean you're going to have your mom's chromosome for height. You might have her finger lengths, you might have your dad's height. You might have your mom's eyes and your dad's hair. You might have, or you might have your mom's hair and your mom's eyes and your mom's, facial features. And this is where we can get sort of interesting genetic combinations where cousins can look more similar than even siblings do. So just for example, I look almost identically to my cousin on my dad's side. I don't look anything like my dad. I don't look anything like my grandparents. I don't really look like my mom, but I look exactly like my female cousin on my dad's side. I look more like her than I look like my own siblings. And that's just because of the way the independent assortment happens. She and I happen to receive a lot of the same genetic information. All right, and one last thing I want to say, along with this independent assortment, you may see some interesting genetic variations between siblings as well. So, for example, I don't know how many of you out there have done those DNA kits, those Ancestry.com DNA kits. For Christmas one year, my mom got all of us kids DNA kits. So that would be, all of us kids and our spouses' DNA kits. I have 2 sisters and 2 brothers. So they're all 5 of us kids and our spouses. Anyway, it turns out I actually have more genetic information, more DNA in common with my youngest brother than his twin sister does with him. So I have an older sister, a brother who is younger than me, and then twin boy and girl siblings that are the youngest. The twins, they are fraternal twins, of course. One is male and one is female, which means they do not share any identical DNA. They are not any more closely related to each other than they are to the rest of us. And in fact, my youngest brother is more closely related to me or he shares more DNA in common with me than he does with any of the rest of our siblings, or than I do with any of the rest of our siblings. So I'm more closely related to my youngest brother than I am to the rest of my siblings. So there you go. Independent assortment can make for some really interesting and fun genetic variation combinations. Moving on to anaphase 1. In anaphase 1, we're going to see the homologous chromosomes separate. The homologs are going to move towards opposite poles, and at this point the sister chromatids don't separate. We are just going from haploid to diploid. We're just going from a 2n number to an n number. So we're just separating the homologs. We're not yet separating the sister chromatids. Each is still a duplicated chromosome with 2 chromatids. We're going to separate them in our next phase. But at this point, we have reduced our chromosome number from 2n to n. We then need to go into telophase 1. In telophase 1, it's going to look very similar to telophase in mitosis. We're going to just rebuild a nucleus around each of our daughter nuclei. So those daughter cells have 1 duplicated chromosome from each homologous pair. Intrakinesis. Intrakinesis is going to be sort of the middling stage between Meiosis I and Meiosis II. It's not quite full-blown interphase. In full-blown interphase, we're going to grow our cells, we're going to double our organelles, we're going to double our DNA, we're going to double our proteins, we're going to do everything we need to do to get ready for mitosis, or Meiosis I. In interkinesis, this is going to be just a little bit shorter. Interkinesis, we have 2 haploid daughter cells. Each one's going to have a duplicated chromosome of each type. So interkinesis is going to be really similar. We need to grow in size. We don't want our cells to continually to get smaller and smaller and smaller. So we need to grow in size. We need to double our organelles. We need to increase our cytoplasmic volume. But we're not going to undergo DNA replication. This is going to be a really short phase. We're not going to be replicating our DNA. Our DNA is still replicated from earlier. We just need to divide it now. So all we really need to do is grow our cell volume and then move into our next phase of meiosis, which would be Meiosis II. This is going to be the mitosis of 2 haploid cells and then the ultimate gamete formation. Meiosis II looks almost exactly like mitosis. The only difference is that we're starting off with a haploid cell and we're going to finish up with a haploid cell, as opposed to mitosis where we start with a diploid cell and we end with a diploid cell. But really, that's the only difference. In Meiosis II, each haploid cell is going to divide into 2 gametes. We're going to end up with a total of 4 gametes per meiosis cycle. Starting off with prophase 2. In prophase 2, the chromosomes are going to condense. The cells have 1 chromosome from each homologous pair. And metaphase 2, those chromosomes are going to line up right down the center of the metaphase plate. Once we line up down the center of the plate, we are no longer in our homologous pairs. We are all lined up right down the center. Then we're going to undergo anaphase 2. Anaphase 2, just like anaphase of mitosis, we're going to be dissolving the centromere and separating our sister chromatids. At this point, they are now considered daughter chromosomes. They're not duplicated, and we are going to be building up our daughter cells. In telophase 2 and then cytokinesis, we're going to end up with 4 haploid cells, all genetically unique. So we're going to reform our nuclear membrane around our new daughter nuclei, or around those daughter chromosomes, and we're going to end up with 4 genetically unique haploid cells. The gametes are going to contain a mixture of maternal and paternal genes. Here is a slide that is just a fantastic overview of the entire process of Meiosis I and Meiosis II. I would spend some time with this slide. You should be able to put post-it notes over all of the words on this side on the other side, put your hands up, block those out. You should be able to talk through both stages of Meiosis I and Meiosis II and talk through exactly what's happening at each phase. Here we have, prophase 1, we're going to have homologous chromosomes line up. We're going to undergo crossing over events. Here we've got metaphase 1. The homologous pairs are going to line up down the center of the metaphyseal plate. We're going to see independent assortment occurring. In anaphase 2, the homologous chromosomes are going to separate. We're going to pull them to the opposite ends of the pole. So on and so forth. You should be able to talk through each one of these phases and describe what's happening. All right, and that's it for this section.